sense when we're talking about the mechanism in which these particular molecules are involved. You know, as I told you, the immune system doesn't know what's good or bad for you, okay? The immune system can only detect what belongs and doesn't belong. And as we will see, even what belongs and doesn't belong is biased, okay? And sometimes there are errors in interpreting what belongs and doesn't belong. Classical examples, of course, autoimmunity, hypersensitivity, where the immune system reacts against something that it perceives being a threat. It is actually not a threat, but the weaponry that the immune system uses to eliminate that threat is more damaging to you than the threat itself. Okay? And specifically, we talk about that when we talk about autoimmunity and hypersensitivity. Now, conceptually, the immune system has to recognize something as foreign. Okay? Well, how does it know that this thing is not belonging? Okay? Well, obviously, you have a repertoire of molecules that recognize what belongs and doesn't belong, and then process it. Okay? So, especially in the acquired immune system, where you have T cells, which are the immune officers of the immune army, okay? Those are the guys who decides how to direct an immune response. And B cells, which also not only act as antigen-presenting cells, but also make antibodies. And the interactions among all these cells have to follow specific patterns, which include somehow coming to some conclusion that something is foreign or not belonging. So, without further ado, what I usually do is show you a couple of pictures. So this is the first picture. This happens to be a friend of mine many years ago. He's a professional photographer and a medical illustrator. His wife is also a professional photographer. And every year, they have kids now, this was a long time ago, and every year they send me these tongue-in-cheek Christmas cards, okay? So look at this picture very, very carefully. Now, second picture. First picture. Second picture. Now, if you look very, very carefully, you start to discerning subtle differences. Okay, for example, the bride is not as pretty, okay? And of course, this is silly and we all laugh, but the reason is why this means nothing to you. This, on the other hand, evokes a different response. Why? Well, because if you go back to your mom and you say, this is my medical school professor in drag, she's going to say, well, you better ask your money back, okay? This is my wife. She's a physician. She puts up with a lot of crap, as you can imagine. Okay? The whole idea is this is very silly. Now, the idea why this is very silly is because you've taken this picture, and once again, you've broken it down into its components. Okay? You just somehow process this picture through some sort of filter that in this case is your experience. Okay? And while this picture means nothing, this picture is ridiculous, okay? Because these are two professionals. You don't expect them to do this, okay? This is a hairy guy in a pink suit, okay? So once again, even though this is all silly, your immune system goes through the same principle that has some sort of bias repertoire, just like your experience, takes an antigen, breaks it down, analyzes its components, jives those components with some sort of molecular mechanism that is inside, okay, and then reassembles it in a certain way in some sort of conceptual function. Now, I'm not implying that the immune system has conceptual function, but just like your conceptual recognition somehow comes to some cellular and molecular conclusion stating this does not belong in what you think of a professor and a physician. Okay? So how does this work? So here are some facts of life. In general, antigens that are uptaken by phagocytic cells and antigen-presenting cells tend to be more immunogenic, tend to be better immunogens, better stimulators of the immune system than antigens which do not go through that process. Okay, now, incidentally, since both T and B cells react to antigens, they do recognize for entities, but in different ways. Okay, for example, T cells from a person immune to a microorganism, so therefore capable of recognizing the microorganism, do not bind directly to that microorganism. Okay? However, they will bind to antigen-presenting cells such as macrophages that have ingested that organism. Also, this works also not only in particulate antigens such as a bug, but proteins too. Okay, T cells do not bind protein antigen in solution, even though those proteins can elicit an immune response. The T cells, which direct that immune response, don't bind to the whole protein. Okay, so the concept came that T cells, which are critical for an immune response, must recognize an antigen in a different mechanism that where the antigen appears in its first appearance within the system. Okay. Now, what are antigen-presenting cells? There's all kinds. Okay. The major classes are monocytic macrophages, B cells, and most importantly, Langerhans dendritic cells. Okay, dendritic cells, once again, and as I said in a lecture on innate immunity, are the major link between the innate and the acquired immune system because they act as antigen-presenting cells. So these are the cells that present antigens to a T cell. And once again, I said this in the previous lecture, the way it works is that the soldiers in the field that detect an enemy, they capture that enemy, they kill the enemy, they break it down into pieces, and then they come back to their officers, the T cells, and present pieces of that enemy to the awaiting T cell. Okay? Once again, here's the guy's head. Here's a piece of his uniform. Here what he was carrying on his uniform. Here's a piece of his flag. The T cells, now being the officers of the army, do two major things. Very important point to remember. First of all, they recognize the antigen-presenting cells as member of their own army. And this is done through the MHC. I'll talk about this in a minute. The second thing that they do is they agree with the antigen-presenting cell. Yes, indeed, I agree with you. By the pieces that you brought me, I agree with you that this is the enemy. Therefore, now, I'm going to send out orders to other cells to amplify the response to this particular antigen. Okay? So the two things that the T cells are reacting against are the antigen-presenting cell identity, which is recognized via your own MHC, and the pieces of that antigen. Now, how antigen-presenting cells internalize the antigen depends on the antigen-presenting cell itself. For example, mononuclear phagocytes or macrophages have various different receptors on the surface that can either recognize an antigen because it's covered with antibodies, and the cell can recognize the Fc portion of an antibody attached to a particular antigen, or by other nonspecific receptors such as toll-like receptors and others. B cells recognize antigen and internalize it by their surface immunoglobulins. And remember, we talked about surface immunoglobulins when we talked about antibodies. And then dendritic cells, once again, the major link between the innate immune response and the acquired immune response, recognize antigens by swooping them in, okay, via pinocytosis. All these internalize antigens and break them down. Okay? So this, in turn, leads us to a concept of antigen processing. Antigen processing, just like your conceptual processing of that silly picture I showed you in the beginning, involves the biochemical events that activate various different T cells, either CD4 positive or CD8 positive, we'll talk about these in a minute, after an antigen-presenting cell binds and internalizes a particular antigen. Now, this, in turn, creates the following concept and what cells recognize what in an antigen. So, in general, when you make an antibody response to a particular antigen, you have to make those antibodies to determinants of that antigen that appear in the form the antigen is first seen. Okay? So these determinants are displayed on the protein antigen in its natural state. On the other hand, so B cells which react to an antigen have to be able to recognize the antigen in its natural state, not a process, not taken apart state, but natural state, the state in which the antigen appears when it first enters the system. So, and this is a very important point, B cell clones that are reacted to the antigen must exist and be selected before chemical processing of that antigen. On the other hand, T cells recognize an antigen or a native protein that has been denatured, that has been chemically processed and taken apart. Okay? So B cells can recognize an antigen as it appears when it first enters the system. T cells can recognize an antigen only after it has been processed. Once again, B cells, dendritic cells, macrophages recognize an antigen as it appears in the battlefield. T cells can only recognize an antigen after that enemy soldier has been broken into pieces. Okay? Now, as far as T cells are concerned, there are various types of lymphocytes, but the major ones that react to antigen presentation are CD4 positive and CD8 positive T cells. Now, once again, you have to excuse me, this is a very simplistic view of T cells because of the restricted nature of this course. But in general, CD4 and CD8 positive T cells are identified because they have a molecule called either CD4 or CD8 on their surface. So CD4 T lymphocytes are generally what they're called helper T cells. And what they do is they recognize antigen that have been associated with class II MHC molecule. Write this down. CD4 goes with class II MHC molecules. And in fact, the CD4 component of the T cell that makes it a CD4 positive T cells acts as a ligand for MHC class II molecules. Once we get to the diagram of how that works, it'll make a lot more sense. CD8 positive T lymphocytes, on the other hand, are cytotoxic T cells. These are like the commandos of the immune army, and they recognize molecules that are associated with class I molecules. Okay? CD8 goes with class I. Now, class I molecules are associated with all nucleated cells, and usually antigens that are presented in association with class I molecules are usually antigens that come from within. They're endogenous antigen. For example, a viral component into a virally infected cells. And the CD8 T lymphocytes recognize the expression of that foreign component of an infected cells along with MHC1 and kill the cell that expresses that particular component. And this is to prevent the cells from spreading the infection. Okay, so CD4 goes with MHC2, CD8 goes with MHC1. So what about the actual physical presentation? Now we'll talk about presentation to helper T cells. So the major step is the uptake of the antigen from an antigen-presenting cell, and then the biochemical processing of that antigen by various different enzymes, different chemicals. And then, once that antigen has been biochemically processed and taken apart, you take the pieces of the antigens and you associate them with class II MHC molecules that are inside the antigen-presenting cell. Okay? Now, at the same time, the antigen-presenting cells also secretes molecules that stimulate the stimulation of CD4 positive T cells. Okay? Once again, depending on the antigen-presenting cell, you have different surface components that allow that antigen-presenting cell to internalize the antigen. Okay, macrophages, you have Fc receptors that recognize an antigen covered with antibody, or you have certain receptor for carbohydrates, or other components that recognize the antigen. B cells, on the other hand, as I said, preferentially take up antigen by immunoglobulin receptors, surface immunoglobulins. And, okay, I have a missing slide. That's okay. So now, this is a very important point, okay? Put an asterisk next to this. An antigen-presenting cell, in order to present pieces of antigen to an awaiting T cell, first has to take those pieces and jive it with MHC components of the cell. Okay? So the better the piece of antigen combines with one of those MHC components, the better that antigen presenting cell can present the antigen. And the better that presenting cell can present the antigen, the better the T cell can respond to that antigen, which in turn results in a better response, overall immune response to the antigen itself. So the association between a piece of the antigen that has been processed and MHC2 molecule is a critical step in order for an immune response to react to a particular antigen. So, and this is what I just said. In other words, the T cell response to a particular immunogen is dependent on whether immunogen contains components capable of interacting with MHC class II molecules. So if the piece of antigen jives well with an MHC molecule, then it can be presented well and there's a good response. If that piece of antigen doesn't jive well with a particular MHC component, then presentation is not as successful or absent at all, and therefore T cells cannot respond, and an immune response cannot be amplified against that antigen. Now, unfortunately, now remember that I told you when you talk about T cell receptors, and when you talk about immunoglobulins, the part of the T cell receptor and the part of the immunoglobulin molecule that reacts to the antigen are continuously randomly made. Okay? So you're constantly making random keys. On the other hand, your MHC repertoire, meaning the MHC components that are capable of interacting with any piece of antigen, are limited by what you got from mom and dad. Okay? In other words, your repertoire is large, but is not continuously made anew. Therefore, there is a possibility that because of your genetic makeup between mom and dad and what you got from your parents, you could have an MHC repertoire that simply lacks that particular specificity of MHC that is capable of reacting with that particular piece of that particular antigen, and therefore your immune response to that antigen is not as strong or cannot be mounted at all. And as a matter of fact, it has been proposed that some populations may be more susceptible to certain pathogens because of their genetic makeup and because of their MHC. Fortunately, however, okay, most cases, not only your MHC repertoire is very large, there's multiple alleles, there's multiple polymorphic genes, okay, but the way that a piece of antigen reacts to an MHC molecule is also promiscuous, meaning that different antigen can bind to the same binding site of an MHC, because MHC recognizes mostly chemical motifs, certain patterns, such as charge or hydrophobicity. So even though your repertoire of MHC is fixed, not only you have a large number of potential specificity of MHC that can react to pieces of antigen, but even within the reaction, the particular MHC component can react with more than one piece of antigen. So different pieces of antigen can stimulate a particular MHC specificity. And of course, that's good. Now, where does this all occur? When I'm talking about peptide, I'm talking about the piece of antigen. The reason why we say peptides is because most antigens tend to be protein or proteination in nature. So a peptide is simply a piece of protein. Okay, so we talk about peptides. Where does the association between the piece of antigen or the peptide and the MHC occurs? Well, it occurs specifically intracellular organelles. Okay, and depending on whether this is MHC class I or MHC class II, they occur at different sites within the cell. So antigens are taken in then, They are processed by various different cytoplasmic proteases, various enzymes, various chemicals that break the antigen apart prior to the transport into the rough endoplasmic reticulum, where, among other things, class 1 and 2 molecules are also synthesized and assembled. Okay, so the MHC peptide association is thought to occur somewhere in the endoplasmic reticulum. Okay, now the difference, and again, I'll show you a picture, it'll make perfect sense in a minute. Okay, the difference between class 1 and class 2 is that class 2 molecules stay in the endoplasmic reticulum plugged up by an additional chain called the invariant chain, also abbreviated LI. Beats the hell out of me why it's abbreviated LI. That's the way it is. Okay, I don't make this stuff up. So the class 2 LI complex is transported into an endosomal or lysosomal compartment where it is swapped with the antigen peptide. Okay? So both class 1 and class 2, I'll show you the picture in a minute, both class 1 and class 2 are transported to the Golgi where they segregate. Class 1 go directly to the surface. Class 2 arrive in a lysosomal-like compartment, and what happens is the antigen peptide is then swapped with the LI, and then the complex is presented to the surface. All right, so here is class 1. Here is the inside of the cell. Okay, here is the endoplasmic reticulum. Here's the Golgi. You have some sort of endogenous, antigen piece, okay, that is broken apart and made it into peptides. Okay, then the peptides are transported into the endoplasmic reticulum. Here is class 1 molecules with a beta-2 microglobulin. The peptides are then associated with the binding site, the peptide side of the class 1 molecules, and then this whole complex is brought to the surface where you have a CD8 positive T cell, because this happens to be class 1, that A recognizes class 1, and B recognizes the antigen. Now in this case, because this is an internally generated antigen and therefore suggests an aberration of the cell, there's something wrong with the cell, for instance a viral infection, the CD8 positive cells, this is obviously not to scale, okay, kills the cell that presents this antigen. This cell is telling a cytotoxic T cell, I am infected. I have something endogenous that comes from within, okay, because I'm showing this to you via the MHC1 molecule. Please kill me before I spread it to other cells. Now, remember that the endoplasmic reticulum contains both class 1 and class 2. So in a case of a class 2, here is now an external antigen. For example, a phagocytosed microorganism or an internalized protein. This is now broken apart by various different enzymes, and pieces of this particular antigen are brought into what is called an endolysosome. Okay. At the same time, here is the endoplasmic reticulum where you have class 2 molecules with an invariant chain blocking the binding site. Now, why do you want to block the binding site of class 2 molecules? The reason is because you may have endogenous antigens that are binding to class 1. You don't want them to bind to class 2. Okay? So now, this particular complex, the MHC invariant chain complex, is brought into a vessel where parts of the exogenous antigen are also bound, and now the invariant chain is taken off from the binding site, and the piece of antigen is attached to the MHC, and the complex goes to the surface where you have now a helper T cell. So in this case, this particular antigen presenting cell is telling to the helper T cell, listen, here is a piece of enemy, okay? You recognize me because you recognize my MHC-Cas2, and I'm showing you a piece of the enemy that I just captured in the field, And the helper T cell now says, okay, I recognize you as a member of my own army because I recognize MHC class 2, and I also recognize the piece of the soldier you just killed. I agree with you this is the enemy. And so now this helper T cell sends signals back to the antigen-presenting cell as well as to other cells, in this case to amplify the immune response against anything that has this particular antigenic determinant. Okay, and this is the difference between class 1 and class 2. Now, in addition to the MHC components, the T cell receptors, and the CD4-CD8 molecules, there's a host of other molecules that catalyze the interaction between antigen-presenting cells and T cells. Okay? And once again, I don't have the time to discuss all these because, I mean, we have a whole lecture just on these molecules. Okay? But first of all, you have the CD4 molecules that makes the T cell CD4 positive that acts as a ligand for class 2. Okay? And this molecule plays a role in cell-to-cell interaction. The two cells have to physically interact with each other, as well as signal transduction, sending signals inside the cell so the cells know what to do. And of course, you have CD8 in the case of MHC1, which act as a ligand for class 1 molecules, and it too play a role in cell-to-cell adhesion as well as signal transduction. But it is not there. It doesn't end there. There's a whole host of other molecules which are also involved in interactions between an antigen-presenting cell, such as a B cell or a macrophage, and a T cell. And as a matter of fact, these molecules are also involved in many other cell-to-cell interactions. So here is a diagram, and again, no, I'm not going to ask you, does this CD28 binds to B7, okay? It's beyond the scope of this course. But just know that when two cells are interacting, there's a host of molecule either on the surface of the cell or secreted that sends signal from cell to cell, either the cells that are interacting or other cells. So here is an antigen-presenting cell that presents a piece of antigen, a processed piece of antigen that has been associated with a class 2 molecule. Here is a helper T cell that recognizes the MHC class 2 via CD4 and also recognizes the piece of antigen presented by the antigen-presenting cell via the T cell receptor. Okay? So now is the antigen-presenting cells and the T cells are agreeing that this dude here is part of the enemy. So now there is an activation, and further molecules are involved. For example, molecules such as adhesion molecules that maintain the cells in physical contact so that this interaction can occur. And in addition, there's a whole host of molecules, mostly cytokines, we'll talk about cytokines in a different lecture, which are the radio signals among cells of the immune system that go either from the antigen-presenting cells to the T cell or from the T cells back to the antigen-presenting cells. And these are examples of many different cytokines, and each of these cytokines may have a different effect on the cell that it receives it. Okay? Now, these cytokines also instruct other cells of the immune system once this interaction has been achieved. Okay? So this is a very, very complex system that not only involves initial recognition of an antigen and its pieces, but also amplification of the immune response by the action of other molecules, both on the surface of the cells as well as secreted. Now, what happens then when you turn on a T cell? A T cell, once again, especially helper T cells, modulate an immune response. Those are the guys that actually make, and again, unquote, decisions of what the response is. What cells to activate, what part of the army it is to turn on. Okay? So there is an activation stage where an enemy is presented to a T cell now has to respond and instruct its own soldier to do something. Okay, sir, there's tanks out there. Okay, we'll send out the choppers or anti-tank, artillery, or whatever. Sir, there's a wino at the door. Okay, we'll send Joe with a baseball bat. Okay, the point is the T cells have to respond in some way. Okay, and what happens is that they have also additional accessory molecules, which in turn activate the T cell further, and in turn the T cell now responds. Okay? And what the T cell does is does a lot of different things. First of all, it secretes cytokines. It sends out chemical signals to other cells to achieve whatever the response is necessary for that particular antigen. Okay? And of course, it also proliferates. It makes daughter cells, which in turn further expand the secretion of cytokines in order to give orders to the other cells to do something about the antigen. And the sequence of events is, of course, early signal transduction that allows the communication between the outside of the cell to the inside of the cell, activation of various genes. Activation of various genes. If you want to turn things on, you have to turn the genes that express those things. Surface molecules, secretion of cytokines, and increased mitotic activity. These are all things that a cell undergoes when it's activated. The other thing that it does, it communicates with other T cells to do the same thing. Now it calls other officers of the army to do the same thing. And this is done by secreting certain cytokines such as IL-2. Okay, IL-2 then differentiates cells into different types depending on whether, for instance, you have a cell-mediated response that is needed or an antibody-mediated response. Okay, now if you think these are not important, when we talk about immunodeficiency, certain form of immunodeficiency, if some of these cytokines are not capable of being produced, or if the cells that receive the signals cannot receive the signals, you have very, very severe immunodeficiency. We'll talk about the bubble boy in a different lecture. Now, what about B cell activation? B cell activation goes through the similar things as T cell activation. Okay? However, B cell activation initially is activated by the antigen in its natural state. So a B cell can recognize an antigen even in the absence of activation of T cells. And as a matter of fact, it does that because B cells, just like macrophages and dendritic cells, can also act as antigen-presenting cells. Okay? So it does the same thing as any other antigen-presenting cells. It internalizes the antigen, degrades it, okay? It presents peptide in class 2 hemolytic to T cells, and in turn, this is a very important point, receives signals from T cells. And after that, B cell proliferates and goes through the secretion of antibodies. Okay? So a B cell can recognize an antigen when it first appears on the in the system, okay? And it presents it to a C cell. But then it needs a response from a T cell in order to further be activated. So here is a particular antigen or epitope that is internalized, okay, and then is presented to helper T cells using the same mechanism I just discussed. Now, the most important thing to remember, and this is true of many cells, is that B cells recognize antigens by their surface immunoglobulins. However, what you need to do, and this is true of many receptor-ligand interactions, you have to have cross-linking of receptors in order for the cell to be activated. What does that mean? What it means is that if you have a surface molecule, such as an immunoglobulin on the surface of a B cell or a T cell receptor, in order to avoid that something just simply tickles that molecule and activates the cell, okay, you have to have an antigen that brings together more than one receptors for that antigen. This is think of it as a safety mechanism, okay? If you think of a modern power tool, okay, like a rotary saw or a modern pistol, what happens is the power tool or the pistol is activated by pulling the trigger. However, in modern tools or pistols, you usually have a thumb safety button, okay, that you have to depress before you press the trigger in order to activate either the gun or the saw. Okay? This is to prevent something from accidentally hitting the trigger and activating either the gun or the saw. So you have two things you have to press into a rotary saw in order for it to be okay. Well, the same principle applies to cells. Okay, you have to have, I don't know if I have a picture or not. Yes, you have to have a antigen cross-linking more than one of these antigen receptors. In the case of the B cell, this is a surface immunoglobulin. And what happens? What happens is that, remember that a plasma membrane is not a styrofoam ball with things sticking out. It's a fluid sea of proteins, okay, that are capable of moving about. So what happens is that when you cross-link these two guys, there are signals within the membrane that now tell the cell, this is a real thing. This is not somebody just touching the receptor. This is two or more receptors brought together, which in turn bring together various different molecules within the cell. And now the cell knows that this is a real antigenic situation and gets activated. Now, B cell activation takes place in germinal centers. Okay, and then there's a whole bunch of surface molecules that are involved in B cell activation. Okay, and what they do is upon presentation of antigen, what they do is they undergo rapid division and mutation of various genes, and the cell is turned on. Now, the big thing here is, and it's a very complex mechanism which I will skip because once again it's beyond the purpose of this lecture. The key to remember is that B cells that have bound to an antigen and then have been activated and have responded to a T cell are now allowed to survive for a long time. Instead of undergoing through apoptosis, okay, and die off and making new B cell, that B cell that has been activated by a particular antigen and that has to be further amplified by a T cell, it's kept around, and that is a memory B cell. Okay, once again, I use the analogy of a key and lock mechanism. Okay, if you found a key that eventually works in a lock, you made this key randomly, now you don't throw that key out, you keep it in your pocket. So then if that lock comes around again, you have a key ready for it. You don't have to fumble for 1,000 keys. And this is the same thing of what memory B cells are. Okay, they are B cells that have been activated by an antigen the first time they have seen it. They have been further amplified by T cells, and now they're kept around and make daughter cells for a very long time. So then when that antigen comes back, now you have the B cells, this is also true of T cells, that are ready for that antigen to be recognized again. Okay, so the memory B cells basically depends on prevention of dying of the B cells that have recognized a particular antigen. And once again, it's a complex mechanism, but just remember the principle. Now, so if these particular, remember that the repertoire of molecules such as immunoglobulin and T cell receptor is made randomly. Okay? So because you're constantly making random specificities, randomly, you have the possibility that those specificities of the molecule that recognize something as foreign could recognize your own tissue. Because they're made randomly. So how do you deal with that? Of course, you don't want the immune system to recognize your own tissues. And this is done through a concept called tolerance. Okay? Immunological tolerance is basically instructing a very powerful mechanism, such as this, not to react against a specific antigen. Okay? And while you may think that that occurs randomly, it does not. There is a process by which your body instructs or prevents, in most cases, your immune system from reacting to itself. Now, obviously, it's not perfect because if it was, we wouldn't have autoimmunity. So the process by which the immune system is prevented from reacting against itself is a process that involves, unquote, the screening of T cells from birth to maturity. And this takes place in two major mechanisms: positive selection and negative selection. Okay? Positive selection is developing T cells are presented by specialized cells with their own MHC components. Okay, remember that your repertoire of the ability of recognizing an antigen is dependent on your MHC repertoire. Okay, so T cells that are supposed to recognize an antigen, they have to be able to recognize MHC because MHC is the part that presents the piece of antigen to T cells. So the first wave of selection is T cells are presented with MHC. If they recognize MHC, if they bind to it, they're allowed to survive. If they don't bind to it, then they're allowed to die. And this restricts the repertoire called MHC restriction. Now you have a wave of T cells that you have selected that are capable of recognizing MHC. Then you have negative selection. Once you have a repertoire of T cells that are capable of recognizing MHC, you basically booby trap them. What you do, you have specialized cells that present these maturing, these growing T cells with self-antigen. Okay? And if the T cells recognize self-antigens, they're killed off. So you have basically immune system undercover cops, okay, that look for, drug buyers, and what they do is they present, here's a dime bag. Well, if you buy it, you're screwed. Okay? This is the same principle. Okay? Where you have specialized cells that present this MHC-restricted T cell with self-antigen. And if the cell takes the bait, those T cells are killed off. So here is a summary of the of immunological tolerance, central tolerance. So you have precursive T cells. First wave is positive selection, where these guys that are growing up are presented with either MHC class 1 or MHC class 2. Okay? Now, if they do recognize MHC class 1, they're allowed to survive and then become CD8 positive T cells. If they recognize MHC class 2, they're allowed to survive and they become CD4 positive T cells. If you don't recognize, they're simply allowed to die off. Now you have what is called an MHC restricted repertoire. You have a selection of T cells which are capable of recognizing MHC. Now comes the second wave, and the second wave is the presentation, the booby trapping of T cells which react to your own antigens. Okay, and what they do is if they don't interact, they're allowed to continue on, but if they do interact, they're killed right off. So the final results is a repertoire of T cells that A, are capable of recognizing MHC, okay, so MHC restricted, and B, they're not reacting to your own proteins, to your own epitopes. Okay, now unfortunately, this thing is not perfect, okay? This thing is not perfect because it all depends on what particular self-antigen is presented, how well the cell responds to that self-antigen. So there are holes in the system in which a potentially self-reacting T cells could escape. Now, what about B cells? How do you prevent B cells from self-reacting? Well, remember I told you that this is a very important point. A B cell can recognize an antigen in its natural state, but it needs permission, it needs orders from a T cell to amplify its response to that antigen. Okay, so in general, even though you may have self-reactive B cells, because in this particular case, you eliminated the T cells that are capable of recognizing self-antigen. Even if a B cell presents a self-antigen, there ain't no T cell that's recognized as an antigen. You eliminate it in this step, okay? So in general, in a very simplistic way, okay, B cell tolerance is really mostly achieved. I say mostly. There are situations in which this doesn't apply, and once again, we'll talk about this when we talk about immunopathology, okay? Most cases, B cell tolerance is achieved by T cell tolerance, because if you don't have the army officers that are capable of recognizing a particular self-antigen, there ain't nobody to give orders to the B cells to recognize and react to that particular self-antigen. Okay, so this is different type of processing, okay? If you have any questions, if not, next week we'll talk about effector mechanisms.